So we think that the very first type of cell was a prokaryotic cell, or very similar to a prokaryotic cell, and probably pretty close to what we now call a bacterium. So bacteria are pretty simple, even though they've been around for an immense amount of time on our planet. But that immense amount of time on our planet that they've had has led to a huge diversity of bacteria. And there aren't many places on Earth that you won't find any bacteria. They've been found miles down in the ocean crust, right? So while, yes, we think that the first type of cell was a prokaryotic cell, early in life's history, though, a group of bacteria started to kind of follow a different path and diverged off from the rest of the prokaryotes. And this is what we now call archaea. And archaea are kind of a group that are between bacteria and eukarya that have some features of eukarya and some features of bacteria. And some archaeans are thought to have branched off to evolve into what we call the first eukaryotic organisms. And eukaryotes, which we'll start tackling later, but eukaryotes are much more complex than prokaryotes. And that's one of the major distinctions between archaea and eukarya. So I like this picture because it gives us a sense of scale of a prokaryote. So compared to us, they're tiny. There's no way we can see them with the naked eye, right? Most prokaryotic cells are just 1 to 2 micrometers long, and if the average strand of hair is about 100 micrometers in diameter, we could fit 50 to 100 bacteria just across that diameter of hair. So they are way smaller than eukaryotic cells, like the plant and animal cells that we've been seeing under our microscopes. So the average plant and animal cell comes in at right around 50 to 100 micrometers. While their prokaryotic cells are not as small as the virus, they're about the size of an organelle inside of a eukaryotic cell. One of the major ways to classify prokaryotes is based on their shape, their physical morphological shape. So most prokaryotes only come in 3 basic shapes. They come in spherical spheres, it's redundant there. They come in spherical shapes like those balls that we see on the left, and we call those cocci. In the middle, we see these kind of rod-shaped cells. They're kind of a longer oblong shape. We call those bacilli. And on the right, we see the spiral cells. Those are called spirilla. So most prokaryotes are going to come in one of these 3 shapes. So even though there are millions of different species of prokaryotes, they come in one of 3 forms. So that's our first level of classifying prokaryotes, is what shape are they? In addition to shapes, bacteria can be categorized based on whether they live alone, singularly, or if they live in chains, if they connect together to form chains, or if they live in aggregate and they form clumps or colonies. So as we talked about before, prokaryotes have a pretty basic cell structure. They're basically just a sack of cytosol with some DNA in there and some ribosomes. But they do still contain all the major structures that make a cell, right? They have a cytoplasm, they have a plasma membrane, and they have DNA and ribosomes. But what's weird is that prokaryotic ribosomes are actually smaller and less complex than what we see in eukaryotes. And it's actually some of these differences in ribosomes that let us break up bacteria, archaea, and eukarya into 3 different domains of life. And these differences in ribosome structures are also the reasons why antibiotics work on bacteria, but they don't kill our cells. And they don't kill archaean cells either, because they have differences in ribosomes. And differences in ribosomes is one of the main pieces of evidence to support the theory of endosymbiosis for the development of the first eukaryotic cells. So all prokaryotes are surrounded by a plasma membrane, and that membrane can be decently sensitive to environmental changes, but for the most part, all prokaryotes have a layer of stuff, mostly some starches, maybe some proteins that are surrounding the outside of that plasma membrane that we call a cell wall. So outside the plasma membrane, typically we have a cell wall, and the main jobs of that cell wall in prokaryotes is to support the shape of the cell, protect the cell, and resist osmosis. So prevent too much water or too little water coming into or out of the cell, which would cause the cell to burst. So both bacteria and archaea have a cell wall, but the structure can be different. The bacterial cell wall is made up of a molecule called peptidoglycan, which is a complex carbohydrate that forms chains and then can be attached to protein chains. So it's a combination of protein and carbohydrate. The wall of archaean cells lacks peptidoglycan. We'll talk about archaean cell wall structure later, and we'll get back to the cell wall in just a second in prokaryotes in general. So we'll talk about the archaean cell wall at a later point in time. And we'll revisit the cell wall of bacteria in just a second, but let's talk about some of the other characteristics that are unique to prokaryotes. So in addition to a cell wall, prokaryotes can, but don't always have, a few other structures. Some prokaryotes might have a capsule, and a capsule is a dense layer of a polysaccharide, which is a sugar, a carbohydrate basically, or a protein outside of the cell wall. So this is an additional layer outside the plasma membrane, outside the cell wall, that helps this prokaryote, this bacteria, attach to other surfaces. It helps the prokaryotes attach to each other. The capsule might also help protect against dehydration or attacks from the immune system of their host. The capsule might also have these fimbriae, these hair-like structures that extends the surface of the cell. So it could increase the surface of the cell, which is helpful for transporting materials or collecting organic molecules from the environment. But we really think that this capsule is really there for protection and for attachment to surfaces or other cells. Also on the outside of prokaryotic cells, we'll often see flagella, usually one or multiple flagella, and these flagella are basically just proteins that are attached to little motors. They can power these flagella to flip back and forth to allow the prokaryotic cell to move. So approximately half of all prokaryotes are able to carry out directional movement, or what we call taxis. So taxis is directional movement to or from a stimulus. Oftentimes that stimulus is light. It could be some sort of organic molecule, but they can move directionally. They can move to or from a stimulus, and that flagella let them move in that direction. And they can actually move pretty fast. They can move up to 50 micrometers a second, which is 50 body lengths of a prokaryote. In bacteria, the flagella are much smaller than the flagella that we've been seeing on some of the organisms that we've been getting in our moss or a lichen, if you saw any organism with flagella there. These flagella are made exclusively of protein rings that kind of turn in a corkscrew fashion, and they are not covered by a plasma membrane. Whereas in eukaryotes, typically the flagella will be covered by a plasma membrane. Okay, so more on the external structure of prokaryotic cells. One of the pretty common ways to distinguish between different prokaryotes is called a gram stain. So a gram stain is a protocol when used that turns either a bacteria purple if it's gram positive or pink if it's what we call gram negative. And the reason why the stain works differently or turns different bacteria different colors is because some bacteria have an external cell wall structure that allows it to interact with that gram stain. So in gram-positive bacteria, they have a layer of peptidoglycan that is on the outside of the bacteria that can be exposed to the stain and grabs up the stain and then turns the bacteria purple. In the gram-negative prokaryote, they have an extra lipid bilayer outside of their peptidoglycan layer, and that prevents the gram stain from reaching the peptidoglycan. So that purple washes out, that purple gram stain washes out. So gram-positive bacteria turn purple when stained with the gram stain. Gram-negative bacteria remain pink because they have this extra lipid layer outside of the peptidoglycan layer. Okay, so let's zoom in a little bit and take a closer look. So here we're looking at both wall types. So on the left, we see that the plasma membrane of the cell itself is covered by a singular layer of peptidoglycan. So in the gram stain procedure, this is what picks up that stain and turns the cell purple. On the right, we still have a cell wall and we still have that peptidoglycan layer, but it's covered by a second outer membrane. And so during the gram stain procedure, a gram-negative bacteria, that second outer membrane layer, protects that peptidoglycan wall from interacting with the gram stain, and so it doesn't turn purple. So it actually will turn pink, but because we treat it with a second stain, but we'll talk about that later. So we can use this gram stain to classify bacteria, but archaeans lack peptidoglycan, so they will always appear gram-negative. Okay, one more thing about prokaryotes and their cell structure is their genome. So a genome is all the DNA found inside of an organism, all of its genes, and all of the genes that it uses to regulate its genome. And in prokaryotes, their genome is a single circle of DNA. We still call it a chromosome, but their entire genome is a circle. Typically, that main chromosome, which is what's shown here in kind of that bluish-purple color, is located in a region that we call the nucleoid. And it's that one single circular chromosome tightly bundled together with some proteins. But it's not surrounded like a membrane like we see in eukaryotes, right? So it's not contained inside of a nucleus. That single chromosome contains hundreds to thousands of genes. Not as many as we see in the hundreds of thousands of genes in eukaryotes, but it still contains at least thousands of genes. So their main chromosome is a giant circle that's bundled up in the nucleoid region. But some bacteria will often contain DNA in extra pieces that we call plasmids. So these plasmids, like we see kind of here in this red, are small circles of DNA that carry genes with supplemental functions. Often, things like antibiotic resistance will be found on these plasmids. The plasmids are usually free-floating in the cytoplasm and can be transferred between cells in the process of conjugation. But usually the plasmid only keeps around 5 to 6 genes present on that plasmid, so it's not nearly as much as what's contained in the main genome. And when prokaryotes reproduce, they reproduce by a process that's very, very quick called binary fission. Sometimes binary fission can happen within 20 minutes. So it's basically just one prokaryotic cell making a copy of all of its DNA and then dividing into two. So each daughter cell gets an exact copy of both the plasmids and the main genome, that main circular chromosome, in the daughter cell. So if this takes only 20 minutes, it doesn't take long for one bacterial cell to make millions of copies of itself. So that's the end of our introduction to prokaryotic structure. So now let's talk about some of the diversity of prokaryotes.